right, I'm gonna run through what you need to do for the pipe cleaner lab. You're gonna need 2 sets of pipe cleaners and a pair of scissors, preferably the old pair because you're gonna be cutting the wires and we don't wanna ruin our good ones. So the first thing you're gonna do is we're going to cut these pipe cleaners into our chromosomes. We're gonna make 2 cuts so that we end up with 3 pieces of each pipe cleaner, 1 small, 1 medium, and 1 large. And you wanna make sure that you can easily tell them apart. So it's kind of easier to cut these one at a time. So here's all of my small chromosomes. Okay, then I'm gonna cut the medium ones and the large. Okay, so when we start, we're gonna just start with our diploid cell that has 1 copy, of each one, like this. Okay, so we have 6 total chromosomes here. Then during S phase of interphase, these are going to replicate. So I'm going to show that now by putting on the matching pipe cleaner, which now these have become sister chromatids. Each of these are an exact copy of each other. This is still a diploid cell. It still has 6 chromosomes, but each of the chromosomes just has a copy. So then we're going to go through mitosis. The front page of the pipe cleaner lab is mitosis. Remember that the purpose of this is to replicate the cell. We're going to start with that diploid parent cell we saw, and we're going to end with the 2 diploid daughter cells. So first we're going to do prophase. Prophase is where all of the nuclear membrane is going to dissolve. The spindle fibers are going to start to move, and centrioles move to the poles, and the chromosomes are going to condense into these visible shapes by supercoiling. Okay, then metaphase is going to occur, and they're all going to line up along the equator of the cell, just like this. We'll just line them all up. Okay, so notice that in mitosis, they just line up by sister chromatid. They don't form those bivalents where they associate with their other homolog. We just have 1 row, so that in anaphase, we see separation of the sister chromatids to each side as they're pulled by spindle fibers, like this. Okay, and then eventually we get to telophase, where the chromosomes are going to decondense. So that's kind of hard to model because I don't really know how to decondense a pipe cleaner. The nuclear membrane reforms, and now we have 2 cells that are identical to what we started with, each with 6 chromosomes. That's mitosis. So now let's contrast that with meiosis, which is on the back of the pipe cleaner lab. So we're going to start with the same thing. Okay, 1 cell that's got 6 chromosomes, right? Each of these, one's from mom, one's from dad. So you have 1 copy from each of the parents. So again, the first step is exactly the same, where all of these are going to be duplicated in S phase of interphase. So now we have sister chromatids again. Okay, but what's special is that in prophase 1, okay, in prophase 1, these bivalents are going to form through synapsis. So we're going to have a set of sister chromatids that's going to line up right next to its same size set of sister chromatids. So the one from mom and the one from dad. Remember, these have the same genes at the same locations, but they might be different forms of the genes called alleles. So what's special that happens at prophase 1, I'm going to use this one as the model, is that crossing over can occur. And when that occurs, there can be an exchange of genetic information. Now I'm going to show that by cutting a piece of the end of one of the chromosomes. Whoops. Like so. That's touching this one. And you're welcome to twist these together if you'd like. I'm just crossing them. But I'm going to twist this to show you that they're exchanging genetic information. So now there's just a little piece of that chromosome there and a piece here. Okay, so that's an example of crossing over that's occurred here during prophase. Okay, and you could do it in all of them if you wanted to. Then we're going to get to metaphase. All of these are going to meet in the middle. They're going to line up like this. Notice this is very different from mitosis where they all just lined up in one row, and so the sister chromatids could separate, because remember that in meiosis 1, we have separation of these two sets of sister chromatids, this homologous pair. So then anaphase, we're going to see separation like this. Okay, and then telophase, it's going to decondense. Now we have two cells, but remember that meiosis has two divisions. So then we're going to show the second division. Prophase, let's go into metaphase. Now I'm going to show these, hopefully I'm going to move those over. These are happening now in separate cells, so there's two different cells here, pretend. Okay, then anaphase, where we see separation now of the sister chromatids, like such. Okay, and so our final product at the end is going to be 4 cells, each with half of the genetic information. And notice that they're different, crossing over has occurred. Oh, I didn't really mention random orientation, which I should have, which was earlier in metaphase, that there was a 50-50 chance of which side during metaphase that each of these lined up on, okay? And that occurs both in metaphase 1 and metaphase 2, random orientation. So maybe the one from dad goes this way, maybe the one from dad for this one goes that way, right? So depending on which side of this plate these line up on, right, it's going to split in very different ways. So you're going to have different gametes every single time. Okay, hope that was helpful.